everyone, this is RUSSA here and I'm going to talk to you very briefly about my research topic today. Imagine you walk into a room and you forget why you came in. I do it many a times. But what if that confusion didn't fade after a few seconds? Now, that's what life is like for those who are living with Alzheimer's. It is a disease that erases memory. You tend to forget names, misplace your belongings. And today it's not rare. Nearly millions of people are affected worldwide. But what causes this disease? So over a century ago in 1906, it was first described by a German neuropathologist, Dr. Alois Alzheimer, who was studying his patient, August Dieter, who was suffering from memory loss. What he found was something strange. He saw some sticky clumps and tangled fibers inside the brain, which are now called as amyloid beta plaques and neurofibrillary tangles, the two hallmarks of aging, and the leading cause behind the degeneration of neurons. Now, what actually causes these plaques? Now, there is a protein on the cell surface called APP, which when cleaved wrongly by two cutters, they generate amyloid beta fragments. So what happened to these fragments after they are generated and how they communicate inside? So that brought us to a special class of proteins, RTKs or receptor tyrosine kinases. Now you can imagine those as cellular antennas sitting on the surface, receiving messages from outside and transmitting them into signals inside. Now this signaling is very important for cell-to-cell communication, their growth and survival. But misfiring of these receptors or responding to wrong signals may lead to disaster. Now there are several classes of RTKs, but what caught our attention was DDR2. Or discoidine domain receptor 2. Why? Because it belongs to a unique set of RTKs that normally responds to collagen, and it is unexpectedly upregulated in AD where collagen expression is very less. So that made us question, can amyloid beta be the unexpected signal or trigger behind DDR2 activation in AD? Apart from that, we also looked in Alzheimer's like cell that DDR2 is not only present, it is also interacting with amyloid beta inside the cell. That made us wonder what happens next. So DDR2, which is a traditional membrane receptor, actually travels inside the nucleus, the heart of the cell in Alzheimer's-like condition. Not only that, inside the nucleus, there are some of these tiny structures called nuclear speckles. You can imagine those as factory where some splicing events takes place. So there, they interact with some splicing proteins, and together they regulate the alternative splicing of base 1 gene. Now, if you imagine this gene as a movie and if you edit it differently, it generates different versions of the story. So same thing is happening with base 1. The active isoform is being generated, which in turn is cleaving this APP protein, which I showed earlier, leading to the generation of amyloid beta. So DDR2 here seems to receive wrong signals from amyloid beta going inside the nucleus and rewiring gene expression in AD. But further studies are need to follow up this pathway. Till then, thank you so much.